Hi everyone, my name is Skylar Osborne, and in this presentation I will be explaining how light moves from the retina to and through the brain. There's a lot that happens throughout this process, so let's jump right in. The visual pathway begins with the retina, so we'll start there. The retina is made up of 3 layers of cells. There's the photoreceptive layer, which is the deepest layer, then there's a ganglion cell layer, which is the shallowest layer, and lastly there's the bipolar cell layer, which acts as a bridge between the photoreceptive and ganglion cell layer. When light enters, it travels towards the back of the eye, through the transparent bipolar and ganglion cell layers, to the photoreceptive layer. This is where sensory transduction occurs. So, what is sensory transduction? In order to understand transduction, first we need to know a bit more about photoreceptors. There are 2 types of receptor cells, rods and cones. Rods are responsible for processing what we see in low light, and cones process what we see in bright or moderate light. Embedded in the membrane of the outer segment of photoreceptors are photopigments. Photopigment molecules are made up of 2 components, a protein called opsin and a lipid called retinol. When light falls upon the photoreceptors and photopigments are exposed to light, they split apart into those 2 components, opsin and retinol. This separation is what triggers a change in membrane potential, also called the receptor potential. So, let's answer that question. Sensory transduction is the process of an external stimulus, in our case light, being converted into an internal stimulus, which would be that receptor potential. Now, what happens after light is transduced or transferred into a receptor potential? An action potential. How does this happen? Well, receptor potentials actually control the amount of neurotransmitter that is released from the photoreceptors, which will end up affecting the rate of firing in the postsynaptic cells. Photoreceptors form synapses with bipolar cells, and remember earlier I said that bipolar cells connect the photoreceptors to the ganglion cells. So, the receptor potentials will cause the photoreceptors to release neurotransmitter to the bipolar cells. They will then fire and release neurotransmitters to the ganglion cell layer. This causes an action potential. Where does the action potential go now? Well, it goes to the optic nerve. The axons of the ganglion cells gather together at the back of the eye into a bundle called the optic nerve, which sends signals from the retina to the brain. These signals cross over the optic chiasm. The optic chiasm is made up of axons from the ganglion cells, and they cross over each other to form an X shape. This part is where it gets a little bit tricky, so listen carefully. The axons of the inner halves of our retinas, which are those closest to our nose, cross over the optic chiasm. So, the axons from the right half of the left eye cross over and continue on to the right hemisphere of the brain, and the axons from the left half of the right eye travel to the left hemisphere of the brain. This means that the information from the inner halves is processed contralaterally. The axons of the outer halves of our retinas do not cross over. Instead, they stay on the same side of the optic chiasm as the hemisphere that they are on. The axons from the left half of the left eye lead to the left hemisphere, and the axons from the right side of the right eye lead to the right hemisphere. This means that information from the outer halves is processed ipsilaterally. Where do the signals go after they leave the optic chiasm? To the lateral geniculate nucleus. Information is sent to the LGN, which is part of the thalamus in the brain. It is made up of 6 layers. The first, fourth, and 6th layer receive contralateral signals, and the second, third, and 5th layers receive ipsilateral signals. The first and second layers are called magnocellular layers, and the 3rd through 6th layer are called parvocellular layers. Ventral to these layers is the coniocellular layer. So, where do the signals go after the LGN? To the striate cortex. Also known as V1 and the primary visual cortex, the striate cortex receives information from the LGN. It is the first cortical region to combine visual information. V1 is made up of 6 layers, and within these layers are 3 different types of cells that process different basic visual mechanisms. And all of this happens in the modules, which we will talk more about later. Now, where does all of this processed information go? To the extrastriate cortex. Information from V1 is integrated and combined in the extrastriate cortex, also known as the visual association cortex, or V2 and above. Visual processing can occur in 2 different streams of the extrastriate cortex, the ventral or the dorsal stream. The ventral stream receives information from the magnocellular and parvocellular layers in the LGN. The dorsal stream receives information mostly from the magnocellular layers. So, that's how light moves from the retina to and through the brain. Now that I've described the visual pathway, we must discuss the types of information that are processed at each stage. Let's begin with the retina. So, color perception begins in the retina and has two steps. The first step begins with the photopigments. Remember earlier when I discussed the two components of photopigments, opsin and retinol? Well, rods only contain one type of opsin, rhodopsin, and this means that they are only sensitive to one kind of light and do not differentiate colors. However, cones have three different types, which are sensitive to different wavelengths. There are red, green, and blue cones, so unlike rods, they do distinguish colors. The second step takes place when photopigments communicate with ganglion cells. The rate of firing determines color perception, and ganglion cells are sensitive to either red and green, yellow and blue, or brightness, and when they are exposed to these lights, they are either excited or inhibited. For example, if a cell that is inhibited by yellow and excited by blue is exposed to blue light, it will become excited and the rate of firing will increase. The retina also helps with depth perception and spatial location. Now let's move on to the LGN. Magnocellular layers process information about form, movement, and depth. Parvocellular layers process information about color and fine details, and although there has not been enough research done in order to be sure, there is evidence that coniocellular layers process some color. What about the striate cortex? Simple cells process information about orientation and location. Specifically, they tell us about edges of lines and objects. Complex cells process information about orientation regardless of location and movement. And hypercomplex cells process information about objects, specifically where a line ends. The striate cortex also has around 2,500 different modules, and each is responsible for a specific area of the visual field. Within the modules are CO blobs. These detect color. Outside of the blob is where everything else is processed. Lastly, let's talk about what is processed in the extrastriate cortex. The ventral stream processes information about form and color. Different areas of the ventral stream are dedicated to different kinds of objects. The EBA is responsible for body parts, the FFA is responsible for faces, and the PPA is responsible for scenes and backgrounds. V4 is responsible for our ability to realize that colors are the same in different lighting. Now, the dorsal stream processes information about motion and spatial location. V5 is responsible for radial, circular, and spiral movement. So, now that we know how light moves from the retina to and through the brain, and what information is processed at each stage, let's talk about a recent study regarding the influence of tint on visual processing. Schilling et al. Hypothesized that color-tinted lenses could produce effects on how we process visual information and how we respond to emotional events. The participants were shown pleasant, neutral, or unpleasant images while wearing no glasses, and again with either blue, red, green, or yellow tinted lenses. The results indicated that viewing neutral images while wearing red tinted glasses produced the same potentials as viewing pleasant images with no glasses. Their findings also suggested that the response to images is modified at the cortical level of processing. Based on what we know about the two cortices involved in visual processing, we can determine that this response to images is modified in the extrastriate cortex, specifically in the ventral stream. This is because the ventral stream is responsible for processing color and combining or associating it with other information in the brain. Thank you so much for listening and good luck on your test.